by this afternoon, so I will check during break, lecture and lab, and see if they're here before you guys are out. Because they came a little early last time, so we'll check and see. And then during lab time as well, if they don't, I'll print off, photocopy a list of the lab structures. But I have your lab and lecture exams to get back to you as well. And we have 2 days on heart anatomy. We'll start a little bit of the physiology. Thursday is the dissection day. So we're doing sheep heart dissection. So the goggles and the gloves and the closed-toed shoes and all that kind of stuff applies, and it should be up on the list. So let me just go through a couple of these slides on the basic, what you're looking for in anatomy, in the embryonic anatomy development. You may remember, in the cross-section of the embryo, there were 2 blood vessels identified as dorsal aortas, and those 2 blood vessels join together in the central region of the heart in this fashion. And this essentially is what you need to know for the embryology of the heart. Okay, is this lecture slide right here. So the 2 dorsal aortas join together and fuse to form a single tubular structure. That part's easy. And then they begin to rotate. And so we find, notice we have 2 vessels coming in and 2 vessels leaving. And this is the beginning of what is a 2-circuit system of the heart. A systemic circuit system and a pulmonary circuit system. And these should be overlapping. So with 2 vessels coming into the heart, we have blood coming to the heart from the systemic circuit, meaning the body, and from the pulmonary circuit. So which of these would be oxygenated and which of these would be deoxygenated? Okay, pulmonary is oxygenated. Actually, probably should draw that in red. That's our typical oxygenated color. And systemic is not unoxygenated because it's still about 40% deoxygenated. All right. So this is coming into the heart. So in other words, this would be venous blood, all right? They're both venous blood. Anytime blood returns to the heart, it is identified as venous blood, whether it's oxygenated or not oxygenated, because it's under low pressure. And that's how we determine whether it's venous or arterial, not whether it contains oxygen or not. But coming from the systemic circuit would be blood from your muscles and for your digestive organs and from your brain that has used up much of the oxygen and the nutrients. And blood that's coming from the lungs has picked up oxygen. Now this is describing adult circulation, right? Because the fetal lungs don't have access to oxygen. So this is the potential circulation and what we have in our cells. Now, arterial blood is coming from 2 different locations. It needs to go to 2 different locations. So we have these 2 vessels coming in and 2 vessels leaving. And so they are going to go to 2 different circuits. Okay, again, they're arteries because you're leaving the heart. This is under low pressure. And this is under high pressure. So the structure of the vessel is different because of the pressure components. So now it's going to go to the systemic circuit. It's going to go to the organs and muscles of the body. And what was in the systemic circuit is going to go to the lungs. So these are now reversed. systemic that was deoxygenated is now going to the lungs to pick up oxygen. And what was coming back from the lungs as oxygenated blood is now going to go to the body. So we have essentially two circuits that overlap. If they do not overlap, it's incompatible with life. Because you would have deoxygenated blood coming from the body to the heart, going back to the body, coming to the heart, going back to the body, and never picking up any oxygen. And you would have blood going to the lungs and coming back to the heart and going to the lungs and coming back to the heart and never delivering that oxygen to the body. So this doesn't work where they're two separate circuits. They have to overlap like this. So it comes to the heart as deoxygenated blood, goes to the lungs, picks up oxygen, comes back to the heart, goes to the body, and so on. Now we had, a few years ago, a teenage 15-year-old came to visit the school. He was a friend of one of the classmates. He had had some behavioral problems and been suspended from his high school and was going to an alternate program. And as part of building up points or credits to get back into high school, he came to visit our class and to share his story. He was a little shy, so his mom really did most of the talking. But he was the first infant born in Sacramento to have that complete separation of pulmonary circuit and systemic circuit. Now that's not an uncommon problem. His unique case was that there was no hole in the heart. Usually babies that are born with the aorta going to the lungs instead of to the body have a hole in the heart, so there is mixing of the blood. And the blood that goes to the body is somewhat oxygenated. They're in the form of blue baby. All right, but it's still, they can live. He had to have surgery immediately because his only source of oxygen was the mother's blood, was the mother's uterus and placenta, and so they had to have a surgical before he was born. They had to have the surgical set up already, so that he was immediately taken from birth into surgery and put on a heart-lung machine. Because once his umbilical cord was cut, there was no way for the oxygenated blood in his lungs to get to the rest of his body. That part was fine. He had to keep having surgery to enlarge his blood vessels. But unrelated to that, remember when we in Bio 430 we talked about hydrocephalus and how CSF can build up pressure if it's blocked? He developed that when he was about 4 months old. And so he had a shunt draining CSF fluid. And because of the shunt, his neurologist would not let him play football. It had nothing to do with his heart condition. That was fine. But it was the shunt, which was a separate issue. All right, so if we look at this, you can see now there's a rotation that occurs, and the atria kind of move up and on top and behind, and there's a rotation. And inside of all of this, we have the walls growing to create the separation of the chambers. It's not just the tube structure, it's the growth of walls separating the heart into various chambers. So these are just, you don't have to know these terms over here, bulbus cordis or those components, but we start with the two vessels. Again, what you need to know is on this sheet right here. Okay? And so here's the folding that we'd looked at towards the end of our first unit. And the heart develops, here's the two dorsal aortas, and the heart develops in this area right here. Blood cells actually come from the yolk sac and migrate into the body. So this is colored. You can kind of see the components that are going to be formed. So but it says by the 4th embryonic week. It's actually the beginning of the 4th embryonic week. So day 21, 22. All right. 21 would be the first of that, the end of that 3 weeks. So then as this rotates around, see how the atrium is on the bottom, where blood is coming into the heart? So if we go back to this view again, notice blood is coming into these vessels. These end up being the superior vena cava and inferior vena cava. And blood leaving stays at the top. So there's this rotation. The atrium ends up being at the top. And all of our vessels, see how that rotates around? The blue is rotating around. And now we have the receiving chambers at the top. So I want to show just a little bit. It's a 9-minute video. Again, I'll put a link up if you want to watch more of it. Just to give you a taste. And there isn't any sound to it. So here's the area that's going to. Make a little bit more sense since last lecture? This is up really high, above where most of the gut would be. Cardiac jelly is what the cardiac muscle forms from. Day 23, the muscle cells are already contracting. These types of images would have been developed from thousands of sections through embryos. They used to study them by making paper or cardboard cutouts from each slide, tracing around the shape of the particular organ they were looking at, and then stacking them on top of each other to get the whole organ, and then doing that again for each hour or day, depending on what animal they were studying. So you'd have 1,500 slides for one particular organ, multiple sections. Draw that out on a piece of paper, cut it out, stack it on top, and get a 3-dimensional view. Okay, so I'll link this up again. Just wanted you to get an appreciation of the complexity that occurs with the folding. But what I want you to know, again, you have this slide in your lecture notes. And this illustrates, in addition to the folding over here, it illustrates how the chambers are formed by the growing in of these muscular septa. Okay. But like truncus arteriosus, bulbus cordis, you don't need to know those terms. I just want the essential fact is the two tubes fusing together to form a single tube that then develops into the structures of the heart. And you have two inflows and two outflows. Now, so we have, before we move on to, go back to this slide here, before we move on to the embryonic, the location of the adult heart, there are 4 chambers in our heart, all right? 2 receiving chambers and 2 pumping chambers. And that's how we're going to go through and describe the anatomy. When I was a senior in high school, we didn't have AP biology then, but my biology 2 course was an anatomy and physiology course, essentially taught on the college level. and we had to do a project. So my project was heart development, and I got fish heart and turtle hearts and cat hearts, dissected out the entire cat dyed blood vessels and embedded them in resin. Only problem with that is it gets really hot as it forms, cools, and we have these different chambers of hearts. So some animals have 2 chambers, some have 3, some have 4. So a fetus really only needs a 3-chambered heart. If you have 3 chambers, how many do you think are pumping and how many are receiving?
1 pumping, 2 receiving.
Exactly. 1 pumping chamber, 2 receiving chambers. So we see that type of a heart. The fetus is in what kind of environment? An air environment or a liquid environment?
Liquid.
Liquid, okay? So we see that type of 3-chambered heart in other animals that are in a liquid environment like fish. Now, if you think about our capillaries, and I know we haven't done blood vessels, but capillaries are the thinnest walled vessels for gas and nutrient exchange. So they have just a layer of simple squamous epithelium. And as they pass through tissue fluid, you've got the plasma of the blood inside, and you've got the fluid of interstitial fluid on the outside. So if we have a capillary, and we have fluid on the inside pushing against the capillary wall, blood pressure or hydrostatic pressure, we also have fluid on the outside pushing back. Okay? We'll bring up this concept when we talk about how lymphatic fluid and edema occurs. Now, in our adult lungs, well, by adult post-fetus, so baby breathing air, all right, instead of having, so this would be in the body, instead of having fluid on the outside, when we have capillaries that are in the lungs, we have fluid on the inside, then we have air molecules essentially outside of us, which is going to create a greater pressure against the capillary wall, the fluid or the air?
Fluid.
The fluid. So in a fish where we have gills, all right, they're receiving their oxygen from their surrounding water, and the blood flows out into the gills, and they've got the lake water or the ocean water or whatever surrounding the gills. So you've got fluid on both sides of that gas exchange. So you just need 1 pressure. 1 pump means 1 pressure. So the 2 receiving chambers are going to be coming from the body, coming from the gills, and then 1 output going to both the body and the gills. All right, so we only need 1 pressure. 1 pump means 1 pressure. So same pressure is going to the body, same pressure is going to the gills. So how does this fit with fetal development? Their lungs, the small alveoli and air sacs, are filled with fluid. The purpose for that is to keep them from growing into solid balls of tissue. Because then when the baby's born, there's no place for the air to go. So the alveoli, the air sacs, need to have air fluid in them to keep them balloon-like and not growing solid. The gas exchange occurs at the placenta, which is still fluid. We have some blood going to the lungs for gas exchange, but we don't have the air surrounding. And so a fetus really only needs the 1 pump, the 1 pressure system. But as soon as that baby takes a breath, it's going to need a 2 pump system. And that's a little difficult to develop in 2 minutes. So they develop in fetal development with a 2 pump system, meaning 2 pressures. Because as soon as they take a breath, pressure in the pulmonary system is going to drop dramatically as those alveoli become filled with air. So if we have, and this happens in congestive heart failure, if we have a pump pumping blood to the heart, to the lungs, under high pressure, or there's some backup so the blood can't flow through the lungs, then what we have is fluid being forced out of the capillary and into the air sacs. And that's essentially what congestive heart failure is. And so the same volume, as we'll see, is being pumped from both chambers in a baby, or in yourself. The same volume is being pumped to the lungs, which is being pumped to the body. We need to have that for our circuit. If it's a difference in volume, then we're going to start backing up somewhere, but under different pressure. The pulmonary system, once the baby takes a breath, and then throughout adult life, it's going to be under much lower pressure than the systemic pump. Yes, Alex?
So if you die from congestive heart failure, is it almost like drowning? Because you...
It's a similar condition to pneumonia. The cause is different in pneumonia. You both have fluid in the air sacs, but from a different cause. All right. So let's start here now with the actual anatomical structures. The heart by location, we went over this in Bio 430. The heart by location is in 3 cavities and 3 spaces, each a subset of another. So what do we call all of the cavities in the body that are not surrounded by the cranial cavity or the vertebral vertebra? What is all of this space called?
Thoracic?
Dorsal is the back,
Ventral.
ventral cavity. Okay, I know that was a long time ago. I just gave an exam on that yesterday at Sac State, so. We have the ventral cavity, and then above the diaphragm, we have the thoracic cavity. And then everything but the lungs and the pleural cavity is identified as what cavity? Mediastinal cavity. I love this without lecture notes. You guys can't look at the answers. You have to get them out of your head. I know it's more frustrating for you, but mediastinum or mediastinal cavity. Now, that again is everything but lungs and pleural cavity. Right? So there are several components to the mediastinal cavity. We have anterior, and this is essentially between the heart and the sternum. And what's going to lie there? Considering the unit that we just finished, especially in a child. Look at the heart model up there on the shelf. Thymus. In the adult, it's going to be adipose tissue. Okay. We have the superior. And the superior is going to considered to be that part that's above the base of the heart. The base of the heart is up here. So we call the aorta and the superior vena cava the great vessels of the heart. So this is going to be above. And we have great vessels. What else do you think is above there? Based on your knowledge of thoracic anatomy. Trachea. Okay. And behind the trachea, going through the diaphragm to the stomach. Esophagus. And also the thymus, if it's large enough. We have posterior mediastinum. And this is more of an exercise in belief. It's abstract. So remember how the parietal pleura are attached to the ribs and the intercostal muscles? if you were to strip that parietal pleura from its attachment, you would find intercostal arteries and nerves in the sympathetic chain ganglion. That's essentially what we call, it's not really a true space, because it's like pulling the wallpaper off and saying, oh, well, there's a, you know, a hair follicle trapped in there from the previous wallpapering activity. So it's really not much of a space, but what we find there would be superficial to the parietal pleura. So intercostal vessels and nerves. And sympathetic chain. Which leaves us with the largest one. In the center of the thoracic cavity, you can see here on cross section, we have the middle mediastinal cavity. Okay. And that's going to contain the heart. It'll also contain the esophagus, but the primary structure is the heart. Want me to read it off to you again? What part are you having trouble seeing? Posterior or? So this says, above the base of the heart, for superior, we have great vessels, trachea, esophagus, and thymus. You have that? Okay, and then posterior, superficial to parietal pleura, we have intercostal vessels and nerves, and the sympathetic chain. So now in the middle mediastinal cavity, we have the heart. And surrounding that, we also would have the pericardial membranes and cavity. So we covered membranes in Bio 430, and we talked about visceral and parietal, and introduced you to the 3 Ps, pleura and peritoneum and pericardium. Now let's pay a little bit more attention to the pericardium. So this is a diagram from Netters. The parietal pleura has been cut from the lungs. You're seeing it attached to the diaphragm, and the visceral pleura would be on the lungs itself. Here's the thymus. Okay, so you can see as it's shrinking, it's not so much anterior, which would be between here and the sternum, as it is superior. Here's the trachea. These nerves that you see right here are nerves going to the diaphragm. Those are outside the heart. And this is parietal pericardium. We're going to draw a layering of that. So in cross section, again, you can see the heart. Here would be the parietal pericardium. Folds back on itself and becomes attached to the heart as the visceral pericardium. So in the lecture notes, this image is present. But I want to take this and draw it a little bit larger. Unfortunately, with our sheep hearts, we may have a few left over. They're becoming quite rare. But about 5 years ago, the U.S. Agriculture Department required butchers, or whatever they're called that kill the sheep, to, even though the hearts aren't sold for consumption, they had to palpate for parasites or growths or whatever. And to do so, they had to remove the pericardium. And there's a little loophole, so we got a whole new bunch of 100 or 200 of these hearts that still had the pericardium for a year or so after that. And then they closed that loophole. And now we get the pericardia removed. And unfortunately, in removing it, they often take off all the great vessels and much of the atria at the top of the heart. So we'll see what we get on Thursday. But usually we might still have enough to have one sheep heart that has the pericardia on it, and you can examine that, because it can be a little difficult to understand the 3 dimensions of that because of the shape of the heart. So I'm going to draw a diagram of the heart, all right, similar to what you see over there on the left. And we're going to look at the 3 layers that focus specifically on the pericardial cavity. I'm not going to bother drawing the chambers at this point. So lining the inside of the heart, we have a simple squamous epithelium. So the dotted lines are simple squamous epithelium. And deep to simple squamous epithelium, what kind of connective tissue do we have? Loose irregular. Now this is thick enough that there will also be some dense irregular. And so this inner layer, which is continuous with the lining of our blood vessels, is identified as endocardium. Endo for meaning inside, and cardium is our root for heart. That's attached to the cardiac muscle, which we'll look at in more detail when we do the physiology of muscle contraction. Remember, cardiac muscle is different from smooth and skeletal. And this is identified as the myocardium. Now remember our pericardial membrane, similar to when we described the pleura and the peritoneum. The heart grows into this structure as if I were to put my fist into a water-filled balloon. And where my fist is in contact with that balloon structure is the visceral layer. And what's not in contact with my fist is the parietal layer. But it's all one large structure, all right, continuous. So here is the fist. And here is the water-filled balloon where it's attached to the heart. Well, actually, let me draw it the other way around because the heart structure is this direction. So where it's attached to the heart is the visceral peritoneum, and then it folds back on itself and becomes, pericardium. And where it folds back on itself, it becomes the parietal pericardium. So we have an enclosed fluid-filled space known as the pericardial cavity. So I'll label this here, and then I'll draw it over here. Parietal pericardium. Visceral pericardium. And pericardial cavity, which contains serous fluid. That's the simple view, all right? That is very similar to what we have with the pleura covering the lungs. And this fluid provides lubrication as the heart expands and contracts. It slides against the diaphragm and slides against the lungs. However, we don't have any body wall that the pericardium is attached to. The diaphragm is the most structural support. And on the sides of the pleura, there is some attachment. So the parietal pericardium is going to be attached here to the diaphragm, and kind of its fibers will blend into the parietal pleura of the lungs. We need to limit the expansion of the heart. Anybody know why? Why do we not want the heart to just fill up with blood as far as it can go?
Your body needs it.
What happens to, when we talked about muscle, remember there was a graph on the contractile force relationship to overlapping of actin and myosin?
You need to have a certain amount of force.
So if it's too much like this, then it's not going to be able to contract and create too much force. If it spreads apart and there's little to no overlap, then there's no binding sites for the myosin heads to grab onto actin. So we need to limit how far that muscle can stretch out. And so to do that, we add a tough outer layer to the parietal pericardium. All right, so instead of just loose, irregular connective tissue, so in our typical membrane, this would be attached via loose, irregular connective tissue. And then out here, we would have loose, irregular connective tissue attaching it to the body wall. To support that and to prevent increased stretching, we add a layer of dense irregular connective tissue to the outer layer. So that makes this a little bit different. Now I'm going to draw this on a larger scale. So we have our simple squamous epithelium. So when we look at the heart, if we open up the chest cavity and open up the pericardium, we look at the shiny surface of the heart, we're going to see this visceral layer of the pericardium attached to the surface of the heart via loose, irregular connective tissue. Damon, we saw it there. And so this layer is called the visceral pericardium. In fitting with our other terms, endocardium and myocardium, this can also be known as the epicardium. So the three layers of the heart wall. But we're not done yet because we don't have our complete sac. This would be what I'm touching on the surface. So it's not what I'm looking at here. Okay? If I open that up, then that's what I see. So in this view, where I'm touching the heart, I am touching the epicardium, or the visceral layer of the pericardium, fused to the myocardium of the heart wall. So now let's wrap this around and complete our sac. So the simple squamous epithelium. Now we have our pericardial cavity, but that epithelium needs support. So we add our loose, irregular connective tissue. So now this is part of our parietal pericardium. It is the serous layer. The green line is secreting a watery fluid. So this layer is going to be the serous parietal pericardium. But now we need to toughen it up so that we have this capsular appearance that we see here. So only to the parietal pericardium, we have a layer of dense, irregular connective tissue. And just that layer, the dense irregular connective tissue layer, is identified as the fibrous parietal pericardium. Okay. And that's what's shown in this image. So this image is taken here, right about here. Yep, at the base of the heart. So here would be the atrium, one of the atria. Here is the endocardium with a surface of simple squamous epithelium and loose irregular connective tissue. That would be this layer right here. Then we have the myocardium, which are the slashed red lines. And now this is our visceral and parietal pericardium with the pericardial cavity in between. So attached to the myocardium, we have the visceral layer, right here, the visceral serous pericardium, attached to the surface of the heart wall. Then it separates to create the parietal. So now it comes off and it's free to create the parietal. We have a continuation of that serous layer, but now it's not attached to the heart, so it's called the parietal pericardium, with a tough outer layer on top known as the fibrous. I want you to include the word parietal pericardium as well. Okay? So for the parietal pericardium, we have a serous layer and a fibrous layer. For the visceral, it's just serous. You want to take a picture of that? Feel free. Now I want to talk about the borders of the heart and its location in the middle mediastinum. And then we'll take a real short break and come back and talk about the chambers of the heart and their characteristic structures. Okay. The heart doesn't much look like a Valentine's heart. It does kind of have a rounded, pointed region, which is the apex, and a base. And normally we think of a triangle with a point at the top. The heart is reversed. Its typical position is found more to the left side of the heart. The left side of the chest cavity. And you'll notice that this right core of the heart is almost vertical. So the vessels coming into the right chamber, the superior vena cava and inferior vena cava, form almost a straight line. And you can see that right here. All right. Unfortunately, when we get our sheep hearts lately, it's been cut like this, so the superior vena cava often isn't even there. So, but that helps to give a anatomical orientation. It's not going to be like this or like this. That needs to be in a straight line. Okay? And that part is located just deep to the sternum. So that the heart doesn't sit right in the center, but sits with the right border just under the sternum like that, and the apex pointing down. So this would be the anatomical position. You can see that this is almost flat, all right, or horizontal. And then so we have a straight line here, almost a right angle here, and then kind of a diagonal at that location. So if you're trying to, that gives you the view. Well, I guess I can leave that one up there. So this would be the right border of the heart. And it's formed almost entirely by the right atrium. Then we have the inferior border. This is what's resting on the diaphragm. And it's formed primarily, this vessel right here is running in a groove called the interventricular sulcus, meaning interventricular between the two ventricles. So all of this right here is the right ventricle. We're seeing about a third of, or 25% of the left ventricle. So primarily the inferior border is the right ventricle. And the left border is going to be the left ventricle. We see very little of the left atrium. And the superior border or base is typically given to the great vessels, which are the superior vena cava, the aorta, and the pulmonary trunk. The anterior surface of the heart, so all of this, is also given to the right ventricle. I mean, you would see a little bit of the left ventricle, but essentially, primarily, we're seeing the right ventricle. And then the posterior surface. So in anatomical position here, you can see this right here is the right atrium, okay, where blood's coming into from the body, head and trunk and legs. Here is that interventricular groove. So all of this is anterior, inferior border is right ventricle, and anterior surface. And now when I rotate it around, all of this is left atrium. So the posterior surface of the heart is going to be left atrium and left ventricle. I can't flip my board around, so I'll just write that over here. So if the heart was in place and you'd opened up the chest and the diaphragm, if you wanted to touch the left atrium and the left ventricle, you'd have to put your hands around in the pericardial cavity to do that. Okay, let's take a 10-minute break here, and we'll come back. We'll go through the chambers and identify characteristic structures. We'll leave the blood supply to the heart for Thursday. That you and I would have. And so it's a 4-chambered heart. We have 2 receiving chambers, which are the atria. So this is going to be the right atrium. and the left atrium. And then we have two pumping chambers. We have the right ventricle and the left ventricle. All right. So let's look at the receiving chambers first. Those are the atria. And they really don't have as much capacity, as much volume as the ventricles do, because most of the time the heart is at rest and blood is flowing through the atria directly into the ventricles. All right. While the heart is pumping, the valves will be closed and the atria will be separated from the ventricles, but usually the heart is at rest more than it is in a pumping situation. So we have the right atria here and the left atria here, and I'll leave another image up on the board. These are showing that this is more of an internal view similar to what I have. So right atria, right ventricle, left ventricle. Okay. So let's describe the atria first. I've made my heart too large here, but we can build it. So the right atrium receives blood from via three different vessels. It's deoxygenated. So maybe I'll rewrite this in the blue. And it's going to receive blood from the head and neck and chest via the superior vena cava. Superior vena cava, and that is from the head, chest, and upper extremities. Then it receives blood from the abdomen and lower extremities via the inferior vena cava. It's only about an inch long in the thoracic cavity, and the rest of it's found in the abdominal cavity. So abdomen, pelvis, and lower extremities. All right, what do you think? What have we left out? Well, I kind of included it with chest, but the heart itself. Okay, so the chest, the lung, the chest wall essentially is what we're talking about. Not the lungs, but the chest wall. And so we have a vessel that empties into the inferior vena cava just about, empties into the heart just about where the inferior vena cava comes in. And this is known as the coronary sinus. It's a large, all the veins drain into this sinus, and then, most of them anyway, and then it drains into the heart here. You'll be able to see that, at least, on our sheep heart. So let me show you a posterior view. Get to it here. Come on. So this is the posterior view. Here's the inferior vena cava, and this is the coronary sinus. So these major veins are draining into that, and then that empties into the heart. So on our heart model, anterior view, we look at the posterior view, here would be the coronary sinus. So in this view, this is Netter's. So a window-like opening, door-like opening has been cut into the right atrium. Here's the superior vena cava and inferior vena cava. So here's kind of a opening of the inferior vena cava, coronary sinus. All right. Both of the atria have this kind of cauliflower-like appearance. So you can see that here on the right atrium and the left atrium. And that structure is called an auricle, like your ear. And if you open that up, you'll notice that while the rest of the atria is smooth, the inside of that auricle has ridges of cardiac muscle. And that's what's visible here. Okay. The rest of the atria is pretty smooth. We just have these ridges of cardiac muscle. And so we'll add that as a characteristic to our atria, but it's present in both. So I'm going to put it down here in the middle. Actually, let me put the vessels that empty into the left atrium, and then I can erase that so I'm not putting too much stuff on the bottom. The left atrium receives 4 vessels, but they all have the same name. And these are the pulmonary veins. And this is bringing oxygenated blood. This is all deoxygenated. From the lungs. So a bit of a conundrum here for some people that are used to thinking of arteries as oxygenated and veins as deoxygenated. Here is the left atrium on the posterior surface of the heart. All right. Here, colored red, because they're emptying in, they're coming from the lungs, are pulmonary veins. But they're carrying oxygenated blood. So they're called veins because they're under low pressure. So anytime a vessel carries blood back to the heart, it has to be under low pressure or it won't flow back to the heart. Pressure doesn't flow, it is not uphill against the pressure. So pulmonary veins returning blood, oxygenated blood to the heart. All right, so the right side, deoxygenated blood from the body, superior vena cava, inferior vena cava, and then from the heart itself, coronary sinus. Right atrium, 4 pulmonary veins, all right lungs and left lungs. So can I erase that list? And then we'll look at characteristics that are shared by both of the hearts. Okay. Again, these are receiving chambers. Okay. They do a little bit of pumping, but their major role is receiving the blood that just passes through the openings between the right and the left atria, the atrial ventricle, and continues to flow. All right, so structures that both of these have in common are going to be the auricles. We have pectinate muscle. So we have a couple of other descriptions of muscle. We have pectinate muscle, papillary muscle, and trabeculae carni. Pectinate muscle are the only named muscles that we, or type of muscle that we associate with the atrium. This ridge looks like a series of cone fibers coming down. This ridge is called the crista terminalis. You'll see that, but it's not on your list of structures to know. So just recognizing this as pectinate muscle. Another structure that is shared between both right and left atria is a closed previous opening between the right and the left. It was open in the fetus, called the foramen ovale. It's essentially as the chambers of the heart walls grow together, a flap overlaps but creates a gap that allows blood to flow from the right side to the left side. The reason for this in the fetus is because the blood that's coming from the right atrium into the right ventricle is going to be pumped to the lungs. It's coming from the placenta already oxygenated. It doesn't need to go to the lungs to get oxygen. It only needs to go to the lungs to supply the lungs with tissue and maintain that circuit. So most of that can essentially bypass the lungs, and that opening allows that to occur. Because the alveolar spaces are filled with fluid, pressure is higher in the lungs before birth and after. And so that pressure, as the blood comes up the inferior vena cava, almost shoots it through the foramen ovale into the left side. After birth, when a baby takes its first breath, that pressure relationship switches, and over about through a period of 3 days, those overlapping flaps grow together.
Is that what happens when they say a baby has a hole in the heart? It doesn't.
That's the most common cause of heart murmur in the first few days. A hole in the heart can be there, a patent ductus arteriosus, or patent foramen ovale would be that. More serious is a hole that develops embryologically in the ventricle. So that's a normal hole that will usually close, but sometimes doesn't. Okay. A friend of mine, a guy that I used to date a very long time ago, we got back in contact via Facebook. He was a preemie. He was born 6 or 7 weeks early, and in the early 40s. He was a bit older than me. And the care at the time was to put them in incubators with high concentrations of oxygen so that their lungs didn't have to work so hard because they were underdeveloped. And that high concentration of oxygen is toxic to the nerves in the retina, and the blood vessels constrict to prevent the high concentration to getting to those nerves, and with the constriction of blood vessels, the nerves die. And so blindness was not an uncommon complication of premature birth. Well, he became blind, but only in one eye. So he was this medical phenom kind of thing. Well, and I knew that. And so when I saw him on Facebook and contacted him, sent him an email, he wrote back and said, well, excuse my typing, I just had a stroke due to a PFO. PFO, yeah, that doesn't make it, so I had to look it up. And it was patent foramen ovale. So he still had a small, at the age of 60-something, a small overlap that was still open, which allowed blood to settle and a clot to form. And therefore, a small clot had been sent to his brain, causing the stroke. But I wasn't thinking of a patent foramen ovale at somebody his age, but that's what it happened. So there can be a variety of locations on Thursday when we talk about problems with the heart. We'll talk about that. So it develops in this wall right here, between an interatrial septum. It kind of has a thicker rim right there, and when it's closed off, this is described as the fossa ovalis. So I'm going to put it as something that's found in both the left and the right side, but it's easier to see on the right side. Now we have some plasticized hearts. I'm going to pass around probably the best one. So here are the two atria up here. This is the interatrial septum, which is the wall between the two. So if you hold this up to the light, you'll see almost an oval or round area that's thinner that allows the light through more easily. And that would be the patent foramen ovale in this one as well. It's not the fossa ovalis now, but it's what would be open in the embryonic fetal heart. Okay, so those are the structures, vessels associated with right and left atria, internal structures that we see with right and left, all right? And now, at the same time, this is something we'll cover with circulation on Thursday, but blood is flowing into the right side and the left side at the same time. if we follow a single red blood cell, it can only be in one chamber at a time. All right, so we can follow it through, but blood's going to flow into both the right and the left atria, through the open valves into the right and the left ventricles at the same time. Then the ventricles will pump blood out both of the ventricles at the same time. All right, so now let's look at, that's the right atrium. Here's an image of the left, so it's not as interesting. You can see the fossa ovalis. All right, it's just not as thick a wall. So now we need to take the blood through the atria, excuse me, into the ventricles. And we have to do that through valves. There are two types of valves present in the heart. Valves between the atria and the ventricles, what I've drawn here, and valves between the ventricles and the great vessels, which I haven't drawn in yet. So let's take a look at the valves. And they are named for the chambers that they are found between. So we have right atrioventricular valve, and we have left atrioventricular valve. There are actually 3 leaflets in the right atrioventricular valve, and only 2 leaflets in the left atrioventricular valve. Because of that, a couple of different names have been used. So the right atrioventricular valve is also known as the tricuspid valve, and the left atrioventricular valve is also known as the bicuspid, the cusp being the valve leaflet. And if you look at the 2 valves upside down, it reminded somebody of a bishop's mitre, the hat, and so it's also known as the mitral valve. Hence the word mitral stenosis, which is a surgical medical term, and this name persists because of the medical terminology rather than the anatomical terminology. It's a lot easier to remember. It's on the right side. It's between the atria and the ventricles. It's the right atrioventricular valve. It's a little bit long to write out, okay? But that's the setup. Now, if we look at those valves in location, here's the right atrioventricular valve. So this is 1, 2, and 3 leaflets. I've only drawn 2 on the board. The other would be in front. You'll notice that there are strands of collagen fibers attached to these leaflets. The opposite end of the strands are attached to small mounds of muscle found in the ventricular wall. So now we're moving into the ventricles. What I want to attach before we look at the ventricle specifically, I want to indicate that each of these has these cords, which are known as chordae tendineae. The orientation of the valve is hanging down into the ventricle. So now let's look at the ventricles, and we'll talk about the muscles that these strings are attached to. Can I erase this? I think you guys have the notes down. There aren't any differences between the right and the left ventricle. There are between the right and left valves, all right? But not the right and left ventricles. So I'm not going to write that as a separate list. We'll just kind of describe what we have for both. So we have the chordae tendineae. And these are attached to papillary muscle, another key muscle. Somebody thought they looked like small breasts or mammary glands. So these arise out of the wall and provide attachment to the chordae tendineae. Now, see, the valves and the muscles, or the papillary muscles, do not close the valves. All right? Remember, the valves are hanging down in the ventricles like this. The chordae tendineae are attached to the papillary muscles, and the papillary muscles can pull on the valves, but they don't push them closed. We have almost a whole lecture on the cardiac cycle and when the valves open and when they close. And it's important that you recognize that what closes and opens the valves is a difference in blood pressure. So a rise in blood pressure or a drop in blood pressure on one side or the other of the valve is going to close or open it. So I said that when the heart is at rest, blood is flowing into the atria, both left and right, and through the valves into the ventricles. So the valves are open. What keeps them open? Where is pressure going to be higher? In the atria or the ventricles?
Ventricle.
In the atria, all right? Then the atria will squeeze a little bit, and then the ventricles will start to contract. There's no opening at the bottom of the ventricles. It's up here at the top. And as the ventricles start to contract and they raise pressure, blood wells up behind the valves and closes them. So it's the rising pressure in the ventricles that closes the atrioventricular valves. Now, kind of like an umbrella in a really windy day, the valves, I haven't drawn them in yet, but the valves in the vessels leaving the heart haven't opened yet, so we have a closed system. Pressure rises really rapidly, okay? And so what we could have is an instance where the atrioventricular valves that are hanging down inside the ventricle like this, as pressure rises very rapidly inside the ventricles, it could push those valves up and turn them inside out and pump blood back into the atria. So the purpose of the chordae tendineae and the papillary muscles is to resist that rising blood pressure. So the papillary muscles adjust the tension of the chordae tendineae to keep the valves from turning inside out. It's the pressure of the blood itself that keeps them closed. Okay? And then the counter force to keep them from turning inside out comes from the papillary muscles. The closure is the blood pressure. So we have chordae tendineae and papillary muscles in both ventricles. And then if you look at the heart that's going around, it's already gone for some of you, but if you look at the plasticized heart that's going around, and I'll just cross this freely. Look at the wall of the ventricle. You'll notice on this cut here, you may not see the chordae tendineae, but you can see that irregular. Go ahead and take it back around. You can see that the surface is very irregular, somewhat similar in appearance, but a little bit more disorganized to the pectinate muscle. And this surface here would be chordae tendineae, papillary muscle. This surface is identified as trabeculae carni. I always think of that Taco Bell ad with the lion growling. Carni means meat. So trabeculae refers to these cords of meat. All right. So again, ridges of cardiac muscle, and they create an irregular surface. If we were to cut along here, we would have this kind of irregular surface of the wall, the myocardium of the heart. and that's the trabeculae carni. So both ventricles will have trabeculae carni. To me, it's a perfect site for blood clots to form. If you've got this irregular surface, and the blood can settle there, and platelets settle out. It seems like a perfectly smooth surface, like the atria have, would be a lot better, but that's not what we have. So, and most of us seem to do fine with that. Here's the left side. So here are the papillary muscles and the trabeculae carni. Now, we've got the ventricles contracting, the atrioventricular valves are closed, blood pressure is rising very rapidly, so now it's going to open the semilunar valves. And the semilunar valves are found in the pulmonary trunk, which is the vessel leaving the right ventricle, and the aorta, which is a vessel leaving the left ventricle. The vessel that leaves the right ventricle is known as the pulmonary trunk. Pulmonary referring to lung. And it divides and forms a left pulmonary artery and a right pulmonary artery. Vessel leaving the left ventricle is the aorta. So it's going to branch and send blood to all over the body. So here, if we look at the heart model, here's the right ventricle. Blood is flown from the right atrium through the right atrioventricular valve, and now it's going to get pumped out the pulmonary trunk to the left and the right lungs. At the same time, blood is going to be pumped out the left ventricle and up the aorta. So these rising blood pressures have forced the atrioventricular valves closed. They will eventually force open the pair of semilunar valves. The semilunar valves have three cusps. They're found up inside the vessel, and they face in the opposite direction. So here are the atrioventricular valves. These are the semilunar valves. So they both have three cusps. And these are like sconces attached to a wall, like three sconces that fit perfectly. So they close when blood flows backwards and fills up these cusps. You'll be able to see them on the sheep heart dissection. So while the atrioventricular valves are hanging down into the ventricles, the cusps are oriented in the opposite direction. Again, they close because of blood pressure. So when ventricular pressure rises and becomes higher than pressure in the aorta and the pulmonary trunk, again, we have a whole lecture on this, that pushes the cusp up against the vessel wall and blood flows out of the ventricles. As the ventricles relax, pressure is dropping in the ventricles. It's now less than what's in the aorta. It's now less than what's in the pulmonary trunk, and blood starts to flow backwards. And as it flows back towards the ventricles, it fills up those cusps and they close. So they have to fit together in that tri. So we have in the pulmonary trunk, we have the pulmonary semilunar valves, And it's always hard to see the aortic semilunar valves because they're right in the center of the heart. The pulmonary semilunar valve and aortic semilunar valves. We have semilunar valve. Again, a couple of different ways to refer to these. I grew up calling them pulmonary semilunar valve and aortic semilunar valve. You may just refer to them as the pulmonary valve and the aortic valve. If you use semilunar, you either need to say left or right or pulmonary or aortic, so we know which vessel you're talking about. Okay, but you can leave out the word semilunar and just refer to it as pulmonary valve or semilunar or aortic valve. All right, so let's go back to a picture maybe that's clearer than what I've drawn on the board. So here's the semilunar valve. This happens to be, wait, let's see if you can figure it out. You think this is in the thoracic, the pulmonary trunk, or in the aorta? There's two clues up here. They're both kind of subtle. Oh, most obvious to me, because I know the anatomy obviously, is the presence of these vessels. The second is the fact that this has two leaflets. So this is the aortic semilunar valve, and as we'll see on Thursday, these two vessels are the very first branches that come off of the aorta. You can see one right here. These are the coronary arteries. So the very first vessels to supply blood to the heart that come off the aorta are those supplying the heart, the coronary arteries. However, we're almost done here. Looking at the anatomy of this, when the ventricles contract, all right, when the heart is actually pumping blood into the aorta to the rest of the body, are the valves going to look like this or not? No. Those leaflets are going to be pushed up against the aortic wall, and what's going to happen to the opening to the coronary arteries? They're going to be blocked. So blood isn't actually, even though they're the first vessels, blood is not going into the coronary arteries when the heart contracts. It's not until the ventricles relax and pressure drops and the blood starts to flow back and closes these valves that blood's going to flow into the coronary arteries. So that actually makes sense. All right? Trish, I want you to be a coronary artery, okay? So put your fingers in between mine. So her fingers are coronary vessels. My hand is the heart. So what happens when the heart contracts to your fingers? I'm squeezing them, obviously. All right? So if we did allow, thank you. We didn't break your finger.
No.
If we did allow blood to flow through here, it wouldn't get very far in the myocardia of the heart because the heart is contracting and pumping. And so even if that was open, blood wouldn't be able to flow through easily because the heart's squeezing all of the vessels distal to that end. So even in the heart tissue, it makes sense that the blood flows best when the myocardium is relaxed, because now those vessels can be open. So the heart gets its blood supply when you're relaxed. Guess what happens to the amount of relaxed time when you're running or exercising and your heart rate goes up? It decreases. The time it takes for your heart to contract stays pretty much the same. There's less rest time. There's less rest time, but your heart needs more oxygen. So there's a bit of a conundrum there. And that's why people who have narrowed heart blood vessels get chest pain, angina, all right, on exercise, because with those narrowed vessels, they're not getting enough flow during the rest time to get adequate blood supply to the heart when it's working. And so that'll typically show up first as chest pain on exertion. All right, we're going to stop here. On Thursday, which you will have the notes, we will pick up this discussion of the coronary arteries and cardiac veins and how the blood flow gets to the heart. Okay? Let's take a break, let's get back to lab at about 9:30. I need to run some stuff over to administration, and then I'm gonna photocopy the lab list for you guys. But what we're working on in lab are the models of the heart. There are 2 different types. There are the ones that I brought out, and then there are these larger ones. They're called Don Jake Condors. If you're trying to look for their key, heart keys are red. There is actually this one's adipose tissue around. They're a little bit larger. They look kind of scary because they have the nerves and stuff on them as well. All right, but the same structures are available. And then there's those plasticized hearts that I've been passing around all day. So I'll get the lab list back to you about 9:30. I need to run over to administration for just a sec. And then while I'm over there, I might check and see actually if the printer stuff is ready.